Welcome, Laszlo. We'll talk about an interesting topic, food. So, all yours.
Thank you, and thanks, Vinod, for the opportunity of being here. And I'm glad that I managed to make the trip. And indeed, Vinod told you, I have many identities. I'm trained as a physicist, but I define really myself as a network scientist. And in the last kind of decade or so, I embrace really many applications of how we can actually make networks work. And today, my intention is really to tell you about one particular application, which is diseases and particularly the role of food. And let me tell you with a story or with an image. So this is an image that my lab designed for Nature last year when the magazine was ready to celebrate the 20th anniversary of the Human Genome Project. And they turned to us to kind of let us do some kind of data-driven analysis to understand what did the Human Genome Project really do to us? How much impact did it have on our life and so on? This is not the first time we engage with nature like that. And one of the reasons we do that is because we also, we have the data. We studied the science of science, that how do you express science through data points. On the other hand, in my lab, I also have designers, and so we can actually turn this data into something kind of intuitive and beautiful and so on. And the purpose is to tell a story, and in this case, we did tell a story, and what you are actually seeing here is how did the cover ended up being designed. And what we ended up doing is that we analyzed how much work has been done on every single gene before the Human Genome Project and after it. And what you see here is that each iceberg, and obviously the language is clear, each iceberg corresponds to a gene. And you have the chromosomes here, like as individual circles, and the genes are positioned where they are in the chromosome. But then the question is, what is the meaning of the iceberg? And we wanted to use the icebergs to show how much was visible before the Human Genome Project ended, and how much was underwater that was not yet visible and had to be unlocked by such a massive project as the Human Genome. So each iceberg has a size, like the base size, that tells you how many diseases are known to be associated today with it. This is P53, one of the most studied genes, the cancer gene, and 150 diseases associated with that. And above the water, what you see above the water is how much research was done, how many research papers focused on that particular gene before the Human Genome Project, and what's underwater, how much happened after that. So really our goal was to show how much innovation really the human genome unlocked and how uneven that was. So there were some genes that are really overstudied and others are generally very, very ignored. But I would like to emphasize that as pretty as this video and picture became and so on and so forth, it really reinforces two fundamental limitations in the way we think about disease and the way we think about the roles of the genes. And first of all, it looks at genes as independent units, right? And that is that they are just kind of sitting on the genome independently of each other, and they are the source of disease. And the second assumption behind that is that if we are able to catalog all genes and their mutations, we'll be able to cure any kind of disease. And what I would like to show you today is that these assumptions are really limiting us. They're limiting science, and they're limiting actually our ability to cure diseases. So let's start with the first assumption, that genes are independent entities. Well, I told you I'm a network scientist, and what I did in the last 15 years in my lab is to focus on what we call network medicine, which is a new subfield in medicine. At Harvard, where I have one of my appointments, we have a division of network medicine that more than 200 researchers are working on. And what they do and what we do together is to acknowledge the fact that proteins in the cell are not independent of each other, but they work together, they interact with each other. And really what we call life is really the result of these interactions. Proteins themselves are not life. Genes themselves are not life. Life and dynamics of the life really emerges through the interactions of how these pieces are really working together. And as a consequence, function emerges from these interactions, and so does disease. Or the lack of interactions often leads to disease. A little bit like the picture I would like to present today is that that it's not enough to have the components of your car, like a typical car has about 6,000 components. You will not be able to fix or build a car just having the components. You also need the wiring diagram. You need the blueprint of how the pieces are connected to each other. And the purpose of network medicine is to provide us that particular blueprint. Now, how does a disease look in the network context? So this was actually one of our first major discovery in this space, is that when you lay this network out of all the known physical interactions that scientists experimentally have discovered, and then you label the genes involved in a particular disease, and here I'm just showing you asthma as an example, you find that they're not randomly distributed in the network, but rather they tend to aggregate in a certain relatively small network neighborhood. And the reason is simple. A disease is typically a lack of function, the breakdown of a function. And the function is really carried by multiple proteins and other molecules that must interact together to actually achieve that function. So therefore, the genes cannot be scattered or proteins all over the network. They have to be localized in the neighborhood where that function resides. And hence, when a disease emerges, it means that something is wrong with that module. We call this the disease module. Something broke down in that neighborhood, and that breakdown is manifested as disease. So why is this interesting? Well, because these are what we call disease genes, whose mutations are associated with the disease. But if you want to actually cure a disease, you typically don't mess with the disease genes. Very few drugs are actually targeting disease genes themselves. Ideally, we should fix the mutation, but there are technologies that are going there, CRISPR and so on, but most cases we don't know that. And rather, if you look historically of all the drugs that are in the market today, most of them don't hit the disease genes, but they hit in the neighborhood of disease genes because they try to fix the problems caused by the mutation downstream. So therefore, we need to find out what are the other proteins who could be involved or could be targeted. And for that, we have a lower set of algorithms, and you see one in action, which we call Diamond, that is really kind of looking at the neighborhood of disease genes and finds all the other proteins who could be involved in the disease or whose manipulation can actually affect the disease emergence itself. Why is this important? One is that this is how you discover the drug targets, not necessarily the disease genes themselves, but those in the neighborhood. And second of all, most of the things that we had to do in drug discovery will have to be limited to this kind of purple neighborhood that I'm delineating here, that I can actually pull out as a separate part of the network and say, anything that you want to understand about disease has to be limited to that particular smaller neighborhood. It's your flashlight that allows you to ignore the rest of the network and focus in the neighborhood where the trouble is. So what network medicine really allows us to do is to really zoom into the network neighborhood where the action is happening, identify drug targets, identify drug repurposing opportunities, that what other drugs could actually be kind of working that we already have in the market and could affect that particular disease, and so on and so forth. So pretty much the whole tool set actually of how we discover new drugs has to be kind of filtered through this particular network picture. And they also offer us a perspective of how do we think about the relationship between diseases, because diseases are not independent of each other. If you get one disease, you're at high risk for the other one. The most common one is the metabolic syndrome, right? There's a group of diseases from diabetes to obesity that often come together. You get one, you're at very risk at the other one. Could the network perspective give us information about that as well? And it does. As an example, what I'm showing you here is that asthma are the purple nodes, and I'm highlighting also the COPD nodes. COPD is another pulmonary disease. It's often called the smoking disease because the smokers have a high incidence, have a high risk of developing it. But you can get it even if you're not a smoker. And has many, many symptoms that are very similar to asthma, but yet it's a distinct disease. And when you look at the proteins involved in the COPD, they are in the network vicinity of where asthma resides. And this is not unexpected. Diseases that are actually very similar to each other the similarity means that they're typically the same proteins, the same genes are involved. There's high overlap between them. And that means that in the network sense, they got to be interacting with each other, they got to be close to each other. So what the network allows us to do is to take not only asthma and COPD, but to put all the diseases into this network space and try to understand the relationship between them. And here I'm showing you just a big picture of like multiple diseases and whether they overlap or not. And in this network space, this distilled network space, if two diseases don't overlap here, they have nothing to do with each other. But those that overlap, they show high comorbidity, they have very similar symptoms, and in general, there's a hope that the same cure actually will work for both of them, so you can actually hope that you will find common targets between them. So if you want to try to discover new drugs, this is an opportunity to kind of understand these comorbidities. Now, this is kind of the network medicine perspective of really changing the way we think about diseases and getting away from the focus on mutations and single genes, which is really what characterizes today's drug development, and starting to see the big picture, starting to see how the pieces interact with each other and how those interactions really affect our ability to treat disease. And these tools that network medicine provides is kind of wide, vast, quite large. It helps us to find disease modules, drug targets, drug combinations. It actually allows us to find two drugs who may actually together be more effective than alone, and can also allow us to think about personalized medicine, because the disease module helps us understand why is it for some of us the drug doesn't work. Well, we have the same module broken, but there are many different ways to break that module. And if the drug is hitting a certain neighborhood and your problem is in another neighborhood of that module, you may not be effective towards your disease. So it gives a framework to really rethink at all drug development. And one of the reasons I guess I'm here today is because we're trying to bring these ideas and this knowledge back to the patients. And so scipher medicine that was mentioned earlier is one of the companies we funded together with Koshla, and Alif is somewhere here in the audience there, right? And what it does actually is develops network medicine-based diagnosis to identify, whether tell you whether a particular drug will work for you or not, and hopefully down the line, if it doesn't work, what else should you take actually in order to alleviate your problems. But network medicine fundamentally is still rooted in genomics and genetics. And what I would like to spend the rest of the talk is to talk about the fact that how limited that assumption is for us. And in order to do that, let's start first asking the question, why do we die? And there are many sources of that in the United States, but here what I'm showing you is the top 10 sources of that according to official statistics, starting from heart disease to cancer, all the way to kidney disease, and so on. And what is interesting, of course, is that there is a massive genomics approach to these. Many of these diseases, or most of them, have a genetic roots. And the question is, how much can genetics explain from the incidence of the disease? That is, could genes alone explain whether you're going to have heart disease, or there are other factors as well? And every time and there are precise answers to that, because from the genomic technologies, we can predict, or we can actually measure how much of the disease incidence can be explained from that genetic variability we see on patients. And every time I saw one of these so-called Jivas and other talks, I was always very surprised that the effect size was so small. That is, such a small percentage of disease incidence could be explained by the genomic signals that we're having, despite the fact that we have close to full genomic signals about the patients. Like, when it comes to heart disease, 10, 20%, and so on and so forth. So when you look at it, relatively 10 to 20% of the disease variations is explained by genomics, and the question is, where is the rest? And every time I would actually ask that at the talk at Harvard, my colleagues would say, well, it's in the environment, right? And then the question is, what is the environment? And particularly the fact that if only 10 to 20% of the disease causation is explained by genomics, why are we spending half of our research dollars in biology on genomics, why don't we spend it where the disease is coming from? So what is that environment? And as I start asking these questions, the answers are obvious to most of you, right? Stress, physical activity, and of course, food, right? And one of the biggest source of disease that we actually encounter is rooted fundamentally in what we eat and how we eat. And I'm going to show you some data to support that, not that I have to really convince you about that, right? The US has more or less a homogeneous genomic background. We're a mixture of lots of different nations. And yet, when you look at the incidence of heart disease around the US, there are clear hotspots in the regions where there's not much of that. And these are differences that cannot be explained by the genomics background. It's clearly something that we do differently in different parts of the country. But my best example, which is also local, is a study that was done in Japanese individuals of Japanese descent. And if you are a Japanese, genomically you're Japanese and you live in Japan, you have 1.6 over 1,000 people a chance to actually develop coronary heart disease. But if you move to Hawaii with the same genetic background, that doubles. And if you go to move here to San Francisco, it goes further up, right? So where is that difference coming from? And of course, that difference is to a high degree actually lifestyle and environmental effects, and particularly food. So the question I started to ask, we spent the last 15 years developing this wonderful network-based picture of how the cell works. How does the food affect this? How can I take the food that we eat and put these environmental effects into our network-based representation? And I said, well, the answer is very easy, right? At the end, food is molecules. Molecules get actually into our bloodstream, and they arrive to our cells and somehow perturb our cells. So let's figure out what's in the food and how they perturb our cells. And I said, oh, that's easy to do, because we know what's in the food. And the reason why we know what's in the food is that about 10 years earlier, we did a study in the lab, which was kind of fun, had nothing to do with health. It was about what food combination tastes the best. So this was a study driven by molecular cuisine about 10 years ago when it was hot, kind of disappeared, but that time was really hot. And there was a hypothesis in molecular cuisine that ingredients taste well together if they share flavor chemicals. What are flavor chemicals? Flavor chemicals are the volatile chemicals that are in the food, who are mostly sensed by our nose, a little bit by our mouth as well, but mostly nose. And what the molecular cuisine chefs have actually conjectured is that you should combine ingredients that share one or two of those flavor chemicals because you would sense them as a good, well-tasting combination. So we said, well, that's easy. There was a database that tells us what are the flavor chemicals in each of the foods. So we created this massive, beautiful network where the nodes are the ingredients of what that goes into the food, and two ingredients are connected if they share one or multiple flavor chemicals. And you can kind of see the different colors correspond to different classes of food. So like wine and so on are red, and fruits are actually up there in yellow. And the fact that they cluster together is, well, all the fruits share many flavor chemicals because they're similar in nature, and so are the wines and so on. So the question was, is molecular cuisine right? And the answer is yes for European food and to some degree American food. Like if you take lasagna, every ingredient that goes into lasagna except salt shares a flavor chemical with the other ingredients. But it's totally not true for Asian food. Like just with the yin and yang principle, Asian food tries to combine things that don't connect on this map, right? So they try to put kind of composite, opposite flavors together. But it was this study that was more than a decade old that I said, well, we know what's in the food, right? We have a database. And then I was in a surprise, because these are only the flavor chemicals. We have many other chemicals in the food that are not listed typically in this database. So I said, well, if we have the flavor, we get that one too. So let me give an example of how food affects health, right? Well, we know, for example, let's talk about red meat, right? We know that red meat can cause many problems, including, from the cholesterol and so on. But here's one pathway that has been worked out very carefully biochemically that if you eat red meat, then there is choline and L-carnitine in red meat, which is then taken by gut bacteria and broken into TMA. TMA is broken by the liver, processed by the liver and becomes TMAO. And individuals who have high TMAO concentration in their blood are 40% more likely to die of coronary heart disease or cardiovascular diseases than those who have low concentrations. So this is a signal that you're at risk at heart disease. But interestingly, the Mediterranean food, which is one of the healthiest diets out there historically, does recommend to eat red meat. So why don't people at Mediterranean have that problem? Well, because they combine it with garlic or red wine or olive oil, which contain allicin and DMB, and those chemicals block the gut bacteria's ability to actually create TMA. And therefore, you don't have an elevated level of TMAO. So this is a pathway that has been worked out by biochemists, a very well-studied pathway. But what is interesting is that, let's say I'm a consumer and I'm curious, which of these chemicals are in my food or on anything that I eat? And that's when I was in for a surprise. I realized that of the 6 chemicals that are in this pathway, there is systematic information about only one in databases. The rest 5, good luck of finding out what food carries them. And then we dig deeper and said, how is that possible? Because we have nutrition science, right, that is all focused about what's in the food and how it affects health. And what we realized is that, yes, nutrition science really has a robust knowledge about 150 nutrients. These are the nutrients that are the energy sources, sugars, starch, and so on. They're providing the vitamins for you. These are the chemicals that you must have. Otherwise, you will not be able to carry on your life, right? Or lack of that will lead to particular disease, vitamin deficiencies, and so on. But when we started actually at a massive map of figuring out what has been detected in the food, now our current database has 135,000 different chemicals. And this is what we call the dark matter of nutrition. Because you cannot go to any database and find out whether your garlic or your apple or your meat would have these chemicals. Humanity lacks that particular knowledge. We know that they are detected in some foods, but we don't know where, how much, and so on. And you would say, and the typical wisdom is that they don't matter, right? Because they just go in and go out, right? They don't engage with the metabolism, they are not energy sources, and so on. I'll convince you otherwise, but let me give you one big picture. We ended up using the computer to read the biochemical literature, and what we learned out that about 620 health effects that are well discussed in literature are connected to nutritional components. Those are very important, indeed, no question about it. But we found 1,274, that twice as many health effects that are connected to non-nutritional components. So really, much of the health effects that food exerts on us is really not contained in the nutritional piece, but rather in this dark matter of nutrition. So one of the things we do in the lab, and we continue to do, is to try to map out what exactly is in the food. Let me just give you an example. So here is Basil. Has about 2,100 chemical compounds in our database. Some of them are flavor compounds, some of them are connected to disease, some of them have other health implications, and the goal would be down the line, and I'm very surprised that we don't have to have this type of database for every single food, not only what's in there and what's not there, but also to quantify that so we can actually understand their health implications. Now, let's dream a second and let's assume that we have that, right? What would we do with that? How are we going to use this information of what's in the food? And that takes us back to network medicine, right? Because the question is, how do these molecules affect health? And the common wisdom is that we eat stuff, we get energy, and we run, right? But it turns out most of these molecules don't affect health through acting as energy sources, but rather they modulate the activities of our proteins, and they modulate health. And let me give one famous example, or something that we studied a lot in the lab, the role of polyphenols. Everybody knows polyphenols are good for you. You eat lots of vegetables and fruits, and you get polyphenols. They have antioxidant properties, and so on. What is less understood is that your metabolism does not process polyphenols, doesn't even see them most of the time. What polyphenols do is that they're broken up again by bacteria, get into your bloodstream, and they bind to different human proteins. And they affect health to binding by activating the activity of those proteins, blocking certain activities, activating other activities. So we ended up developing the network medicine as an example for polyphenols. So we start with this giant network. This is a sub-network of that, and I'm going to just talk about green tea. The active ingredient of green tea is EGCG, which is a polyphenol. And we identify what are the targets. And then we find out what are the diseases in the neighborhood of the targets that may be modulated by this particular polyphenol. And you can see the hypothesis, if a disease overlaps with the polyphenol targets, then potentially could affect that disease. If it's not, then if it's far away, it doesn't. And based on that, we are able to simply predict what diseases would be affected by EGCG. And this is the list that we predicted in the ranking order. And it turned out that top of them were known, and the rest were not known, right? So not only we recovered all the existing knowledge just simply by this network approach, just knowing what the molecule and where it binds and the network approach, but we are also able to make new therapeutic associations. And it goes further than that. We can make specific predictions of how a polyphenol does that. And in this case, I'm going to give you an example of rosmaritic acid, where we predicted that it should have a role in platelet formation, which is a cardiovascular problem. And not only we predicted that it should have a role in platelet formation, but we told that exactly who are the molecules involved and how it's going to do that. And our colleagues, Joe Loscaso and his lab at Harvard has tested that, and their predictions were right on the money. So this is how food affects health, right? It binds to our proteins, and it changes their activity, and through that, it alters the many cellular processes that are in there. And one of the things I'm doing here today is to talk with Chris Hall, who is here, who has just who's just joined as a CEO of a company that we funded, which is now called Nourishing Health, that is trying to take this knowledge again on the market by designing foods that are specific to a particular disease and trying to develop food-based therapies for patients. So now, slowly kind of get into the last 2 minutes, and I want to go back to where we started from, right? And I started from this video, and now you see some of the perspective change that hopefully I was able to bring today, that this video, as pretty and nice it is, it still reinforces that genes are independent objects. And really, what I learned in the last 15 years is that it may be so, but they work together. And if you really want to understand what life is, how a healthy cell works, and how it breaks down, and how you're able to intervene to fix that, you will need to develop a network-based approach. You have to switch your brain over and think this is all about how things are connected and how they influence each other. And we can also step back a little bit and ask ourselves, is this only about the cell? And I would argue as a network scientist, as well as, and I not have to convince you about that, that when it comes to network, we have this unique focus about the nodes, whether the nodes are people in an organization, whether they are the consumers, or whether they are genes, or whether they are computers, or whether they are web pages. But really, the network is not about the nodes. It's never about the nodes. It's about the relationship between them, about the links. So in a way, if you really want to understand any type of complex system, be it a social system, be it an organization, be the cell, we need to switch our focus away from the nodes and start focusing on the links. That is, how do we relate to each other? Hopefully, this conference is a good example how we can further that. Thank you very much for your attention.